All right, so first off, we have on the left in green, topoisomerase, which I can't pronounce because why? So basically, topoisomerase breaks, sevels, and rejoins the parental DNA ahead of the replication fork and relieves the strain caused by unwinding. The whole point is that when helicase unwinds the DNA, it basically stops all the DNA in the front from like getting super tangled and stuff, because that's what happens when you attempt to unwind two things like together like that. Next, we have helicase, which is in purple. Helicase unwinds the parental double helix, which is the things you see here.
After that, molecules of single-strand binding protein, which are lighter purple, stabilize the unwound strand templates.
The leading strand, which is the leading strand template at least, is the white stuff on the top for the higher one, and then the leading strand is the yellow stuff on the bottom of that. It is synthesized continuously in the 5' to 3' direction by DNA polymerase 3.
So, first primase begins the synthesis of the RNA primer for the fifth Okazaki fragment. Here, primase is the light blue thing.
After that, DNA polymerase 3 completes synthesis of the fragments. When it reaches the RNA primer on the next fragment, it will detach and begin adding DNA nucleotides to the 3' end of the fragment 5' and the replication fork.
After this, DNA polymerase 1, which is seen here as a yellow thing, removes the primer from the 5' end of the fragment and replaces it with DNA and nucleotides added one at a time to the 3' end of another fragment. After the last addition, the backbone is left with a free 3' end.
After that, DNA ligase joins the 3' end to the 5' end of the two fragments.